Greetings, curious minds, and welcome back to Vibing Science. Tonight, we embark on a journey that might seem, at first glance, a little unusual. But I assure you, it will open your eyes to the profound secrets hidden in the most unexpected places. Imagine if the discarded remnants of daily life, something as ordinary as ancient human waste, could rewrite our understanding of history. Tonight, we delve into the world of Paleofeces, an astonishing archive of our ancestors' lives, diet, and health. So settle in, get comfortable, and prepare to have your perception of the past profoundly reshaped. And as always, if you find this journey captivating, a like, a share, or a comment always helps us continue exploring these incredible frontiers. Friends, fellow travelers on the grand journey of knowledge, welcome. Settle in, find your comfortable space, and prepare to delve into a chapter of human history you might not expect. Tonight, we embark on a rather unconventional archaeological dig. Forget the gleaming artifacts, the monumental structures, or the delicate pottery shards that typically capture our imagination. Instead, we're turning our gaze and our deepest curiosity to something far more fundamental, far more intimate, and surprisingly, far more revealing: ancient human waste. Yes, you heard that correctly. We are about to explore the extraordinary world of Paleofeces. Now, I know what some of you might be thinking. Feces? Really, vibing science? And my answer, with a calm, unwavering conviction, is an emphatic absolutely. Because these unassuming remnants, these fossilized fragments of ancient digestion, are not merely waste. They are, in fact, an unparalleled archive. A direct, unfiltered window into the daily lives, the dietary habits, the health struggles, and even the migratory patterns of our ancestors, stretching back through the millennia. Imagine, if you will, a perfect snapshot of a meal consumed thousands of years ago. Not just the type of food, but the very seeds swallowed whole, the tiny bones ingested, the microscopic parasites lurking within, all meticulously preserved, waiting for us to unlock their secrets. This is the profound promise of Paleofeces. While often used interchangeably with the broader term coprolite, which can refer to any fossilized animal droppings, Paleofeces specifically points to these precious human relics. They are discovered, sometimes as part of painstaking archaeological excavations or during extensive surveys, often hidden in the most unexpected of places. Arid caves, where the dry air acts as a natural time capsule, are particularly fruitful grounds for these discoveries. But really, any location offering suitable preservation conditions can yield these biological treasures. The sheer richness of information contained within these ancient offerings is truly astounding. Think of it, a direct record, etched not in stone, but in the very material that passed through the digestive system of an ancient human. We can learn about their diet, certainly through the analysis of plant remains, tiny bones, and even the eggs of parasites. But the story doesn't end there. Paleofeces can also tell us about the individual themselves. Advanced chemical analysis allows us to delve even deeper, using techniques like lipid analysis and the groundbreaking study of ancient DNA. And here's where it gets truly remarkable. The success rate of extracting usable DNA from Paleofeces is surprisingly high, often more reliable than retrieving DNA from skeletal remains. This makes them invaluable for tracing genetic lineages and understanding ancient populations. The fundamental reason this incredible analysis is even possible lies in the inherent inefficiencies of the human digestive system. It is not, you see, a perfectly destructive machine. Not everything that enters is utterly obliterated. While much is transformed and broken down, some elements, remarkably, survive the journey. And while not all of this surviving material is immediately recognizable, a significant portion is. For archaeologists, this surviving organic matter becomes the most direct indicator of ancient diets available anywhere in the archaeological record. No other artifact can offer such an unvarnished, immediate insight into what our ancestors actually put into their bodies. The very process that allows for this remarkable preservation is a fascinating biochemical dance known as the Maillard reaction. This complex chemical process, often associated with the browning of food, creates a protective casing, almost like a sugary shell, around the fecal material. This natural encasement shields the Paleofeces from the corrosive effects of the elements, effectively fossilizing them in a way that allows them to endure for thousands, even tens of thousands of years. When researchers eventually retrieve these ancient specimens, they often have to undertake a delicate and precise process. Typically, the Paleofeces are carefully frozen and then meticulously ground into a fine powder. This powdered form then becomes the canvas upon which the detailed analyses are performed, unlocking the long-held secrets within. So, as we begin this journey into the unseen archives, prepare to shed any preconceived notions, because within these humble, often overlooked remnants lies a profound and detailed narrative of human existence, waiting patiently to be told. The story of who we were, what we ate, how we lived, all encoded in the most unexpected of places. It's a tale that promises to be as illuminating as it is utterly fascinating. Our journey into the unseen archives of ancient life naturally leads us to ponder how such a peculiar field of study even came to be. For much of archaeology's grand narrative, the remnants of human waste were simply that, waste. Overlooked, disregarded, and certainly not considered a treasure trove of information. But like so many pivotal scientific shifts, a pioneering mind eventually saw beyond the obvious, recognizing the profound stories hidden within these humble specimens. The rigorous analysis of archaeological feces, or Paleofeces as we now know them, holds a relatively brief, yet remarkably impactful, history when compared to the study of other, perhaps more glamorous, archaeological materials. Imagine, if you will, a discipline born not from glittering gold or monumental architecture, but from the unassuming and often maligned leavings of our ancestors. The individual who truly ignited this scientific spark was Dr. Eric O. Callan. In the late 1950s and extending into the mid-1960s, Dr. Callan embarked on a scientific path that was, to put it mildly, unconventional. He championed the idea that these ancient fecal remains were not mere refuse, but invaluable biological archives. His early work focused on applying coprolite analysis, a term often used interchangeably with Paleofeces, though coprolite can technically also refer to fossilized animal dung, to unravel the mysteries of early Mexican diets. His groundbreaking papers, particularly those published in the prehistory of the Tehuacan Valley, Environment and Subsistence, laid the foundational stones for this nascent field. He demonstrated, with compelling evidence, that by meticulously examining the contents of these ancient samples, one could reconstruct a remarkably detailed picture of what people ate, what plants grew in their environment, and even the parasites they contended with. It was a revelation, offering a direct, unfiltered glimpse into the daily lives of ancient populations, something no other archaeological evidence could provide with such intimate detail. Despite the undeniable promise of Dr. Callan's pioneering efforts, the field of archaeological coprolite studies remained a rather niche topic for some time. It often takes a while for truly innovative ideas to gain widespread acceptance, especially when they challenge traditional methodologies or venture into areas that might, to some, seem unappealing. Few other researchers initially embraced this specialized path, and the discipline lingered somewhat in the academic shadows. Then, in 1970, Dr. Callan's sudden passing left a void. However, the seeds he had planted were not lost. His vital work was picked up and carried forward by Vaughn Bryant at Texas A&M University's Department of Anthropology. Dr. Bryant, recognizing the immense potential that Callan had unearthed, dedicated himself to continuing and expanding this research. Under his guidance and tireless advocacy, coprolite analysis began its gradual, yet steady, ascent from a curiosity to a topic of serious, rigorous scientific study. What had once been a specialized corner of archaeology slowly, but surely began to gain traction. Today, the analysis of Paleofeces and coprolites has burgeoned, becoming an increasingly integral part of archaeological inquiry. These humble remnants have provided truly profound and often revolutionary insights into the evolution of human health and diet, not just in the Americas, but across the globe. Consider one of the most celebrated examples, the coprolite discovered in the Paisley Caves of Oregon. This single, unassuming specimen yielded some of the earliest and most compelling evidence for the human occupation of North America. It demonstrated, unequivocally, that humans were present in that region far earlier than previously thought, fundamentally reshaping our understanding of the peopling of the continents. Such discoveries underscore the power of this discipline, transforming what might otherwise be dismissed as refuse into a priceless artifact capable of rewriting chapters of human history. The reason such detailed analysis is even possible lies in a fascinating biological and chemical reality. The digestive system, for all its sophisticated efficiency, isn't perfect. It doesn't utterly obliterate everything that passes through it. Some materials, be they seeds, tiny bone fragments, or microscopic pollen grains, manage to survive the digestive process relatively intact. And crucially, some of this surviving material remains recognizable, a testament to the resilience of organic matter and the somewhat selective nature of digestion. This undigested, recognizable material is, quite simply, the most direct indicator archaeologists can hope for when attempting to reconstruct ancient diets. No other part of the archaeological record offers such a direct, unmediated window into what our ancestors put into their bodies. But how do these fragile biological remnants endure for millennia? The secret lies in a fascinating chemical reaction known as the Maillard reaction. This same reaction is responsible for the enticing browning of bread crusts, the rich flavor of roasted coffee, and the delicious caramelization of sugars. In the context of Paleofeces, the Maillard reaction creates a protective casing, a sort of sugary shell, that effectively seals and preserves the organic material from the ravages of time and the elements. This chemical armor slows down decomposition significantly, allowing these ancient samples to persist in suitable preservation conditions, like arid cave environments or deeply buried anoxic deposits. Once unearthed, these preserved archives of ancient life require careful handling. To unlock the wealth of information contained within, researchers typically employ a precise and meticulous process. The Paleofeces are often frozen solid, a crucial step that helps stabilize their delicate structure. Then, they are carefully ground into a fine powder. This powdered form allows for an incredibly thorough and detailed analysis, enabling scientists to extract and identify the microscopic and molecular secrets held within. From these often unremarkable-looking samples, an astonishing tapestry of information emerges. The pioneering work of Dr. Callan and Dr. Bryant laid the groundwork, transforming ancient human waste from an archaeological footnote into a central pillar for understanding the intimate details of our shared past. It is a testament to the power of observation, the willingness to look beyond the obvious, and the relentless curiosity that defines true scientific exploration. The story of human history, it turns out, is written not just in grand monuments and epic tales, but also in the quiet, unassuming narratives preserved within the very refuse of our ancestors' daily lives. And the methods by which we unlock these narratives are as varied and ingenious as the secrets they hold. And so, we arrive at the heart of the matter. The intricate dance of deciphering the secrets held within these ancient biological time capsules. How do we take something that, at first glance, seems utterly mundane and transform into a library of human history? The answer lies in a spectrum of analytical methods, ranging from the broadly observable to the infinitesimally small, pushing the boundaries of what we thought possible to learn about our ancestors. At its most fundamental level, the journey of analysis often begins with simple observation. The size, the morphology, the very shape of a Paleofeces can offer initial clues, allowing archaeologists to make an educated guess about its origin. Is it human? Or does it belong to another creature that shared the landscape with our forebears? These initial distinctions are crucial, though as we'll soon discover, even these visual cues can sometimes mislead. But the true magic begins when we peer deeper beyond the naked eye. Imagine taking one of these preserved relics, perhaps carefully excavated from the dry confines of a desert cave or a perpetually frozen layer of permafrost, and bringing it into the controlled environment of a laboratory. Here, a cascade of microscopic and molecular techniques awaits, each designed to unlock a different facet of the past. Visible to the naked eye, even after millennia, are often the more robust remnants of ancient meals, seeds, fragments of small bones, the indigestible husks of plants. These are the obvious breadcrumbs, directly pointing to what was consumed. But a more nuanced picture emerges when we turn to the microscopic realm. Under the lens, pollen grains, those intricate, species-specific dusts of ancient flora, become visible. Each grain is a tiny fingerprint, revealing not just what plants were eaten, but also what flora was present in the environment, even what seasons were being harvested. Similarly, phytoliths, microscopic silica structures formed within plant cells, offer another layer of dietary detail, resilient and distinctive, even when the rest of the plant material has long since degraded. Yet, perhaps one of the most compelling insights delivered by these ancient deposits concerns the health of the individuals who produced them. Hidden within the matrix of Paleofeces, often astonishingly well-preserved, are the eggs of parasites. These tiny biological markers are not just indicators of disease. They're storytellers of living conditions, hygiene practices, and even the migratory patterns of ancient populations. Imagine uncovering the eggs of a particular intestinal worm, prevalent in one geographical region but rare in another. Its presence in the Paleofeces of a distant settlement could suggest trade routes, population movements, or even shared culinary practices that facilitated its transmission. It's a biological detective story, written in the very stuff of life. Now, we move beyond the microscopic into the truly profound realm of molecular analysis. This is where the science truly takes a quantum leap, allowing us to extract information that would have been unimaginable just a few decades ago. Ancient DNA analysis, a field that has revolutionized our understanding of evolution and population dynamics, finds a remarkably fertile ground within Paleofeces. Unlike skeletal remains, where DNA can degrade significantly over time, the success rate of extracting usable DNA from Paleofeces is surprisingly high. This is partly due to the very nature of the digestive process. Not everything is utterly destroyed, and what survives is often encased in a protective matrix. The DNA extracted from Paleofeces can serve multiple purposes. Firstly, and perhaps most directly, it can definitively identify the species that produced the sample. This is particularly crucial when morphological analysis might be ambiguous or when dealing with highly fragmented or degraded samples. Was it human, or a wolf, or an early domesticated animal? DNA provides the unequivocal answer. Secondly, and perhaps even more fascinatingly, ancient DNA can reconstruct entire dietary profiles. Consider the groundbreaking work conducted by Hendrik Pointer and his team at Heinz Cave in Texas. They analyzed fecal samples over 2,000 years old, and through careful DNA extraction and sequencing, they were able to identify not only that the samples belonged to 3 distinct Native Americans, based on mitochondrial DNA similarities to present-day indigenous populations, but also the astonishing diversity of their diet. What they found challenged existing assumptions. Previous theories suggested that these ancient populations primarily relied on berries. However, the DNA evidence painted a far richer picture. While visible remnants of some plants were absent, their DNA sequences revealed the consumption of buckthorn, acorns, ocotillo, nightshade, and even wild tobacco. And it wasn't just plant matter. DNA from bighorn sheep, pronghorn antelope, and cottontail rabbits confirmed a substantial intake of animal protein. In just the roughly 2 days of food represented in a single fecal sample, Pointer's team identified DNA from 2 to 4 animal species and 4 to 8 plant species. This discovery profoundly reshaped our understanding of the nutritional diversity and resourcefulness of this archaic human population, revealing a far more sophisticated and varied diet than previously hypothesized. It's a stark reminder that what we don't see can often be just as important as what we do. Another powerful molecular tool in this archaeological arsenal is lipid analysis. Lipids, a broad group of naturally occurring molecules including fats, waxes, sterols, and fat-soluble vitamins, are often remarkably stable over long periods. Within Paleofeces, scientists specifically look at fecal sterols and bile acids. These molecules vary significantly between different animal species due to distinct gut biochemistries. Just as a human body processes food differently than, say, a dog or a cow, these unique metabolic pathways leave behind distinct lipid signatures in their waste. This allows researchers to distinguish between human and animal Paleofeces, even when other methods might be inconclusive. A compelling example comes from a Neolithic site of Çatalhöyük in Turkey, a place famous for its incredibly well-preserved settlement layers. Here, large mineral deposits, essentially ancient garbage dumps, frequently contain fecal material, sometimes as distinct coprolites, other times as compressed cesspit deposits. Initially, based on the presence of digested bone fragments, it was often assumed these belonged to dogs, which were common companions in Neolithic settlements. However, meticulous analysis of the lipid profiles revealed a surprising truth. Many of these coprolites were, in fact, human. This revelation had profound implications for understanding the daily lives, sanitation practices, and even the social organization of the people of Çatalhöyük, painting a more intimate picture of their interaction with their waste. And then there's the story of ancient migrations and the global spread of diseases. Consider the identification of fish tapeworm eggs within fecal material found in cesspits in Acre, dating back to the Crusader period. This particular parasite was rarely seen in the Levant region at that time, but was notoriously common in Northern Europe. Its presence in Acre's ancient waste strongly suggests that it was transported to the region by the incoming European Crusaders, a biological footprint of a major historical event. It's a silent, almost invisible record of human movement and its ecological consequences, preserved in the most unexpected of archives. The success of all these analytical methods, from the microscopic identification of ancient seeds to the molecular decoding of DNA and lipids, hinges on a fascinating biological principle. The digestive system is simply not perfectly efficient. While it does an admirable job of breaking down food and extracting nutrients, it never achieves absolute destruction. Certain elements, resilient molecules, and even entire microscopic organisms manage to pass through relatively unscathed. And critically, once excreted, these remnants can be preserved through various natural processes, the most intriguing of which is often the Maillard reaction. This chemical reaction, usually associated with the browning of food during cooking, plays a remarkable role in the preservation of Paleofeces. In essence, it forms a protective casing, a sugary shell, that encases the organic material, shielding it from the destructive forces of the elements and microbial degradation. To unlock the information within, researchers often employ a surprisingly straightforward yet effective technique. They freeze the Paleofeces and then carefully grind it into a fine powder. This powdered form then becomes the canvas for the array of analyses we've discussed, allowing scientists to systematically extract and identify the myriad clues hidden within. From the visible seeds that whisper of ancient meals, to the microscopic parasites that chronicle ancient ailments and migrations, to the molecular echoes of DNA and lipids that speak of identity and dietary diversity, Paleofeces stand as an unparalleled direct indicator of ancient human lives. No other part of the archaeological record offers such an intimate, unfiltered glimpse into the daily existence of our ancestors. They're truly the unsung archives of the past, waiting for us to decode their stories, one microscopic fragment, one molecular sequence at a time. The deeper we look, the more we learn, and the more profoundly we connect with the silent, vibrating echoes of those who came before us. We've peeled back the layers of this extraordinary science, from the foundational discovery of what ancient waste truly represents, to the meticulous methods scientists employ to unlock its secrets. Now, it's time to delve into the captivating case files, the real-world discoveries that have not only rewritten sections of our human story, but have often surprised and humbled those who sought to understand our ancestors. These are the moments when Paleofeces moved from a fascinating niche to a pivotal piece of archaeological evidence, offering unparalleled glimpses into lives long past. Consider, for instance, the remarkable insights gleaned from the Paisley Caves in Oregon. Tucked away in the arid landscape of North America, these caves yielded some of the earliest and most hotly debated evidence of human occupation on the continent. And what was this evidence? None other than ancient human feces. These seemingly insignificant remnants, carefully excavated and analyzed, contain preserved DNA and dietary markers that push back the timeline of human presence in the Americas, challenging long-held theories about when and how our ancestors first arrived. The implications of such a discovery are profound, forcing us to reconsider the routes of migration, the speed of human expansion, and the adaptability of early communities to new and varied environments. It's a testament to the power of Paleofeces that it could spark such a significant reevaluation of our origin story in an entire hemisphere. But the revelations don't stop there. Let us journey to Hines Cave in Texas, where a team led by Hendrik Pointer embarked on an astonishing genetic quest. Imagine, if you will, recovering fecal samples over 2 millennia old. 2,000 years, encased in the dry embrace of a cave, preserving fragments of life from individuals who walked the earth when empires were still rising and falling across the globe. Pointer's team, utilizing advanced DNA analysis techniques, managed to extract genetic material from these ancient deposits. The findings were nothing short of revolutionary. Firstly, they confirmed the human origin of the Paleofeces, linking them through mitochondrial DNA similarities to present-day Native American populations. This in itself was a powerful connection across the vast expanse of time. But the deeper dive revealed something even more astonishing, a detailed menu of the ancient diet. Previously, archaeologists had largely assumed that this particular population relied heavily on berries for sustenance. However, the DNA hidden within the feces painted a far richer and more complex picture. The researchers identified DNA sequences of buckthorn, acorns, ocotillo, nightshade, and wild tobacco. What's truly remarkable is that no visible remnants of these plants were present in the fecal matter itself. This highlights the incredible sensitivity and power of molecular analysis, detecting traces that the naked eye, even with a microscope, would miss. Beyond the plant kingdom, DNA evidence also pointed to animal species such as bighorn sheep, pronghorn antelope, and cottontail rabbit. This dietary analysis fundamentally reshaped our understanding of these archaic hunter-gatherers. Instead of a berry-centric diet, it was revealed that in the approximately 2 days' worth of food represented in each fecal sample, individuals consumed between 2 to 4 animal species and 4 to 8 plant species. This wasn't a struggling population subsisting on meager resources. It was a community with an extraordinary nutritional diversity, skillfully exploiting a wide range of available flora and fauna. This discovery offered a vivid, almost personal snapshot of their daily lives, their foraging strategies, and their intimate knowledge of their environment. It transformed a theoretical understanding into a tangible, scientifically supported reality. Moving across continents, we find another compelling narrative at the Neolithic site of Çatalhöyük in Turkey, one of the largest and best-preserved Neolithic sites in the world. Here, vast min deposits, ancient refuse heaps, frequently contain fecal material, sometimes as distinct coprolites, other times as compressed cesspit deposits. Initially, based on the presence of digested bone, these deposits were often attributed to dogs, implying a significant role for canines in the daily life and waste management of the settlement. However, a closer look, utilizing lipid analysis, once again challenged these initial assumptions. Researchers analyzed the fecal sterols and bile acids present in the material. These unique molecular fingerprints vary significantly between species due to differences in gut biochemistry. The analysis revealed that many of these coprolites were, in fact, human. This seemingly simple distinction has profound implications. It changes our understanding of waste disposal practices at Çatalhöyük, hinting at designated areas or perhaps even different societal norms surrounding human waste than previously imagined. It allows archaeologists to differentiate between animal and human activity within these middens, providing a more accurate picture of the settlement's function and the lives of its inhabitants. It's a stark reminder that even the most seemingly mundane aspects of daily life, like waste, can hold crucial clues to grander narratives. Finally, let us consider the truly global reach of Paleofeces analysis, even extending to the study of ancient diseases and human migration patterns. Imagine discovering evidence of a specific parasite, not indigenous to a region, within ancient human waste. This is precisely what happened in Acre during the Crusader period. The identification of fish tapeworm eggs in cesspit samples from this historical site provided a compelling piece of evidence regarding the movement of people across continents. The fish tapeworm was rarely seen in the Levant region during this time, but was common in northern Europe. The presence of its eggs in Acre's ancient latrines strongly suggests that this parasite was transported from northern Europe by the incoming Crusaders. It's a remarkable biological timestamp, a microscopic signpost of human migration and cultural exchange. These tiny, resilient eggs, preserved for centuries, tell a story of soldiers, settlers, and the often unintended biological cargo they carried with them as they traversed vast distances. It offers a unique window into the health challenges faced by these populations and the biological impact of large-scale human movements. These case studies, from the ancient caves of North America to the Neolithic settlements of Turkey and the Crusader strongholds of the Middle East, vividly illustrate the power and versatility of Paleofeces research. Each discovery, each careful analysis, adds another brushstroke to the grand tapestry of human history. They remind us that the most unassuming remnants can hold the most extraordinary secrets, whispering tales of diet, health, migration, and the very fabric of ancient human existence. And as technology continues to advance, as our understanding of molecular preservation deepens, who knows what other incredible stories are still waiting to be unearthed, locked away within the most primal archives of our past. The journey into the unseen archives of human life continues, one ancient fragment at a time. And so, our extraordinary journey into the forgotten archives of Paleofeces concludes. From the pioneering efforts of dedicated scientists to the mind-bending revelations extracted from millennia-old remnants, we've seen how even the most overlooked aspects of existence can hold the keys to our past. It's a testament to the persistent curiosity of humanity and the incredible stories the universe holds. Thank you for joining me on this deep dive. If this exploration sparked your imagination, please consider liking this video, sharing it with a fellow explorer, and subscribing for more journeys into the cosmos and beyond. What were your most surprising takeaways tonight? And what other scientific mysteries would you like us to unravel next? Until next time, keep exploring, keep questioning, and stay curious.